Okay, I want to thank you, all the organizers of this nice event, especially Dr. Maribel and Dr. Gabriela. No, seriously, thank you everyone for being here, for accepting some of the invitations, for submitting abstracts. I think it has been a wonderful conference, at least for me. And I also like to highlight all the support that we got from the ISCB to make this happen. So Seth, Mallory, and Diane have been really helpful. So now I'm going to tell you about a tool that I have developed. Well, we have developed together with a bunch of people here. So especially Toño Ramirez, that he's sitting there. He's the one who has implemented most of the work. This work I have been doing since I was a PhD student, so it has taken a lot of time. But finally, it became real that we could have a tool. And all these people that are here, I will take a lot of time to say their names, but they have been like great collaborators who have put an input to develop REvolutionH-tl. And REvolutionH-tl stands for Reconstruction of Evolutionary Histories Tool. And the TL is because Toño comes from a region from outside Mexico City where they speak the language where many of the words finish with tl, right? So he really wanted the tl, so he insisted that we have to have a tl. So this is how we made it. Okay, so it's called REvolutionH-tl, and I'm going to tell you about how we developed this. So there are tons of mathematics and tons of algorithms behind this. So I'm going to focus on a very small, but very, very small part, which is the best match graphs. So thank you to Betty that already gave some definitions about that. But I'm going to tell you a little bit of background. So the reconstruction of gene families or genome evolution has been important and has been like interesting for many researchers already. And they want to know always like what we have now, what the genomes that we have, the genes that we have, how they are ordered, and how did the genes emerge by duplications, or these orthology relationships between different species, how they can have the same function, right? So this is an example. This is an example of that I found from Said et al. This is the reconstruction of the homicide genome evolution, and it identifies these different trajectories where, like, there are duplications, speciations, gains and losses of genes, and then what they say is that every species have different trajectories. This is another example. This is by Han et al., where they talked to 12 Drosophila genomes, and what they found when they reconstructed the evolution of these genomes, they found that in some lineages there was like rapid evolution. Okay, so evolutionary scenarios, what we know is that these are branching processes, right? Like we have seen in these talks, we have seen a lot of trees sometimes, and we know that we want to represent the evolution of life or the evolution of the species that we see with a tree, and the tree is basically representing branching processes. So how we describe the evolution of trees? So we say that there was an ancestral species, and then there was some divergence, and then until we have two new descendant species, right? So in a species tree, I will call it S, so whenever I say S, this is species tree. The leaves represent the extant species, right? So every leaf has an extant species, and the inner nodes in this species tree represent speciations, right? So we know that speciations are not really like now, right? But for the sake of this presentation, I will represent that like this. So this is a speciation node, this is a speciation node, and they represent ancestral species. But we know that the genes also evolve, so as the genomes evolve, so as the species evolve. So when you see a T, that means this is a gene tree, and the gene tree in the leaves, what we see is each leaf represents a gene, right? Okay, So, but what we know is that genes evolve as species evolve. So we have like a merge, kind of embedding of the gene tree in the species tree, inside the species tree. And we know this is how it should happen, right? Like it cannot be that the gene tree talk one way and the species another way, right? Because they are inside the species. So what we also know is that these genes go through several evolutionary events. I'm going to focus on 3 events specifically here. One is the speciation event that we already said. Duplication events. Duplication events are the ones that give rise to new genes, and gene losses. So, so I'm going to put also the labels of the events in the gene tree. So whenever I see that there was a speciation of the species, so I will also see that the gene that was the ancestral has to be inherited to the 2 new species, right? So if there's a duplication event, then it gives rise to a new gene, so then I have 2 genes now. Same for this side, so here in this case we have a duplication event, so now we have 2 copies, the 2 copies made it to this ancestral species, and now they are inherited, so there was here this speciation event, and the 2 genes are inherited to the new species, but then we know that there are also gene losses, right? So I can see here that this is a gene loss. So I'm going to give you a couple of definitions that we need to understand how this works, but an orthogroup is a set of species, it's a set of genes, sorry. It's a set of genes that descended from a unique gene. Sometimes it is referred as a gene family, right? Because a gene family is a set of genes that descend from a common ancestor. Then we can define, if we already know all these, so we know the species tree, we know the gene tree, we know the labels that gave rise to these gene trees, then we know which genes are orthodox. For those who don't know what orthodox are genes in different species that have very, very similar sequences, and they say they have similar function in different species. Right? So for example, if one gene does DNA repair for example, there is one gene that does DNA repair in humans and it does DNA repair also with tlacoche, for example, right? So those are genes that are orthodox and their sequences are normally very similar. So, and the genes that are paradox are those genes that arose via duplication event. So then I can build a graph where each node in my graph is a gene, and I throw an H between 2 genes if they are said to be orthodox. And I know exactly which are orthogonal because I can see them in my tree, right? So A, the common ancestor, so this was inherited by this speciation, so I know they should be orthogonal, right? But A is not orthogonal to B2 because the lowest common ancestor, or A and B2 is a duplication event, so they are paradox. So they are actually out paradox because they occur before a speciation. Okay, and the same thing I can do for paradox. So I can build as well a graph where genes again the nodes are genes, and I throw an H if the evolutionary event that gave rise to those genes is a duplication event. Okay, so now what is the question? So this looks very beautiful if you see it like this, and you know which genes are orthogonal, which genes are paradox, you even know what genes are close. But in reality, the only thing that we know is that we know the genes because we have already a lot of annotations, and we know the species where they belong to, and we have their sequences, but from the sequence we have to infer everything else. We have to infer the phylogenetic trees, so species tree, gene trees, orthology, assignments, and whatever else. So, so we have developed during my PhD and afterwards postdocs and as a professor, we have developed a theory that tells you, okay, if I have this set of genes and I can estimate certain similarity between sequences, what this is telling me about the evolutionary history of this set of genes. So basically, the question is like if I if I can have an estimation of the gene trees, what I can say, so is there a tree that describes the set of genes and the relationships between them? And when I talk about relationships, I'm talking about orthology and paralogy. So we developed this theory that is called the best match theory, and basically what it says that if you have a gene tree, and genes and the genes are colored in this case with the species. So a species A is in color orange, and B and B1 and B2 are in red color, and C2 are in green color. So they are this is for one species, this is for another species, this is for another species, right? So evolutionary, there are genes that are closer than others, right? Like A is closer to B1 than to B2 in this gene tree, right? So then if I know the history, then I could say that B1 is the best match of A in two different species, right? So then I throw an arrow like this. So I said B1 is the best match for A. But B2 is not the best match because there is another gene that is a best match for it. And so on. And then I will do it for every pair of species. I take the genes and then I say, okay, what is the best match for the species orange and green? Well, there is only one gene and there is only one gene, and there must be a best match, so I will say that C2 is a best match for A1. And then I do that for everything. And then I have this, what we call it the best match graph, and which is a directed graph with the leaf set of these genes. So I'm not going to go into the theorems and the mathematics, but basically, what this best match tell us is that we have informative triples. So if I see that Y is the best match of X, so X is in one species and Y prime is another species. So if Y is a best match of X, but Y prime is not a best match of X, then the evolution of these three genes must look like this, because X and XY are close, and Y is farther, because this is not a best match. On the opposite, if I have a best match like this, so that Y is a best match for X, and Y prime is a best match for X as well, then that means that evolutionary, like the evolution, they must be like farther. So then X and XY are in this cherry, and then they have the same distance to X prime. So with this very simple theory, and we call this informative triples, with this very simple theory, we can approximate gene trees. So what we do is, what we say is that there is a vertex-colored graph, and if this graph is a best match graph, then there exists a gene tree that represents this relationship. So we start then by estimating this graph, and then we see whether we build the gene tree. Okay, so this is something that we have implemented, although this is part of the core. Also, as I said, there are many other characteristics, mathematical characterizations, and algorithms inside here. But basically, what we have a revolution tool. What we did is that we have an input that is a FASTA for each species. You can have a set of genes or the whole genome in FASTA files, and then we compute alignments between them, right? So as Joao does with his method, we also use BLAST or Diamond or one of the fast methods to just align, and based on these alignments, we calculate hits, and then from the hits we build this graph, and then from the graph, basically we know which genes are an ortho group. Then we check whether they are best match graphs, and then from the best match graph, there are algorithms that allow you to go from graphs to trees. So I'm going to walk you through the pipeline. So this is, as I said, those are the sequences. Then we find the best matches. The best matches between the best hits actually first between the different genes, and once you have the matches and we know that this is a best match graph, then we can decompose this graph into triples. Once you have these triples that I already showed you, that are the informative triples, with the informative triples, you can infer the gene trees, and not only that, but you can also infer the labeling of the inner node, so you know which event occur in each node. Once you have the gene trees, you can also decompose the gene trees into triples. And then, with this information, you can also infer species trees. I'm not going to go into all the details because there are several algorithms that you need to do that. But, and the last thing that we do is that once you have a gene tree and once you have a species tree, you can map the gene tree, the nodes of the gene tree onto the species tree. So the leaves, you map them, of course, to the species they belong to. And the inner nodes are mapped to the species tree, so then we can have something that we call reconciliation of the species tree and the gene tree. So this reconciliation allows to merge, let's say, gene trees onto the species tree, and not only that, but during the reconciliation you can also infer gene losses. So these arrows that look like they didn't make it to the end. So those are what we infer. Okay, so I'm just showing one plot to show you the comparison of how good the REvolution tool does in comparison to other tools. This is only one, but we compare with many other tools for every step: orthology assignments, orthogroups, gene trees, species trees, reconciliations, and we do very good and much faster, way much faster than these tools, for example. So some applications that we started. So when we had the beta, let's say the really beta version of this, so that was in 2021 when Toño was doing still his master's, because now he's almost in the middle of the PhD. He implemented this reconciliation in order to have this paper published. So this is we were working together with Alfredo Varela of the Institute of Neurobiology in some gene families, studying some gene families that are important for brain development in mouse. And we found this beta-cadherin, proto-cadherin cluster in the mouse, and we wanted to know whether this cluster was conserved. So we looked at other vertebrates where the cluster was found, and then building with the REvolution tool, this is what we saw. So this is the species tree in green, and then you can see how the evolution was going, right? Like where the duplications arose and the losses, and we also saw the same thing. So that was, and the nice thing is that one of the reviewers, whom we don't know, he actually sent us a note saying like, oh, your tool looks really nice and blah blah, why don't you publish it as well? And this is what we did. We said, okay, thank you. So something that Katia, who's also sitting around there, has been working a lot with the infection of tomato virus, and we also wanted to study what are the gene families that were defending when there was the infection. So we also found that there is very important family that is called BHLH family. For us, was really nice to find this family because we were also interested in intronless genes, so genes with only one exon and no introns. And we looked for this. We also published in a paper that this is a master regulator in the genome, and it has been found in many other genomes. So this is a comparison of the reconstruction of this gene family where we have the BiGEvo gene. Here, you believe me, but this is tomato and this is domesticated tomato. So one of them is domesticated tomato and the other is the wild tomato. And then we also have potato and chilies here. And then this helps us also to go into microproteins, to study microproteins. This is another gene, so this is very different. As you say here, these are all tomatoes here. and this is Chile. And for example, we saw that this BHLH is also a microprotein that is only conserved in Solanacea, in sub-Solanacea, but not in Chile, for example. So this was the first plot. So I just wanted to say that these plots that I'm showing you here, we didn't draw them by hand. This is an output of REvolutionH-tl. So it's hard, like we haven't seen any tool that does that. So Tonio has been working on that a lot. And of course, we wanted to have an application here in Cuatro Ciénegas, right? So when I met Gabby and he told me about the strains that the isolates that he was working on, then we wanted to also understand these antagonism interactions that Betty just talked about, and Gabby has also talked about. First, we wanted just to have a look at the evolution of these 78 strains in terms of duplication and speciations. Here there are some gains that might be horizontal gene transfer, but this is something that we are working on now, on identifying them. And what we aim to is to also generate these visualizations that are like informative for anyone, that you don't have to analyze the data or the tables that REvolutionH-tl gives you before you can see something, right? So now this is a figure that is generated also by REvolutionH-tl automatically. And when you see here, these squares here is the rate of losses, gains, and duplications. And the redder the square is the more duplications that are, and the greener is the less duplications I have. So you can see that there are, like, as you will see for each strain, even though these are all, or almost all bacillota, right, Gabby? So they have different trajectories as well. And here is the change of gene content. As you can see, like most of them lost a lot of genes, but there is one that is really big that has gain more genes. So then, because we started this project of antibiotic-resistant genes, so we decided to do something simple that was taking the CAR database and these 78 genomes and then put them in REvolutionH-tl and see if how much of the gene families that are known to be in microbes, antimicrobial resistance genes that are in the human body, in the microbes, were also conserved in Cuatro Ciénegas. So we applied REvolutionH-tl. And if you want to know more about this, Tonio has a poster that is going to present tomorrow, so you should go see it. But this basically, we see like a lot of our families conserved along the tree, this species tree. And another thing that we started doing with Manuel, sorry that I forgot to put your name, Manuel, but Manuel has been working on specifically looking at those clues of antibiotic-resistant genes in these strains, and not only in these strains, but also in the metagenomes that we have from Cuatro Ciénegas. And something that is nice is that let's say the antibiotic-resistant genes profile of each genome. So each line is a genome, and then this is an R family, reflect very much the phylogeny. So basically, if you only take the arcs, it will give you a clue of what the phylogeny is. And if you want to know more about this, then go and see Manuel's poster, who's going to present as well on Friday. So now what we would like to do is, as I said, we want to know also what genes are gained by horizontal gene transfer, also in this data set. And we have questions like horizontal gene transfer is more common intrasite or intersites, and what are the antibiotic resistant genes which are transferred. And this is something that Tonio has been working in this method. So in the mathematical characterization, we have been working on that, but he's working on a computational method to detect horizontal gene transfer. And if you want to know more, he's going to give a short talk tomorrow about this. So I just want to say that, so this tool was published last year, and we have just released the second version of it. So the paper has been sent to PLOS. And now the tool is even faster than before, and it has more granularity on the gene trees. And so if you want to use it, please feel free. It's free in PyPy. So, and please feel free also to ask us questions on how to use it. Toyo has documented the tool very well, so then there is a data set to also run it as an example, and then you can run it on your data. It's also, we also present them in different boxes because it's also modular. So if you want to use other steps like separately, you can do it. And I just wanted to show you, this is the visualization that we have now. Okay, so I think I don't have to say more, but I want to invite you. So we are organizing the first workshop on algorithms in bioinformatics in Sinvestav, Irapuato, as part of the Ciencia Viva that Gabi organizes. And this is in the last week of July. And we expect we will have like wonderful speakers and also instructors from abroad and also from Mexico and of course from Sinvestav. And if you are someone from the field of life sciences or exact science, but are interested in algorithms, please join this school. And thank you very much.
Thank you very much, Maribel. We have a little bit of time for
like a very short question.
Thank you, Maribel. Yeah, this is great. I was wondering, so like a basic model of what happens when a gene family duplicates or expands is that predefined selection relaxes so that accumulates more mutations, to allow for neo-functionalization. So do you see that when you detect, if you can date it to the specific speciation event, do you see also like a concurrent increase in the mutation rates?
Well, no, we don't look at mutation rates. So, but this is something that you can look at once you have detected your gene families. But something that you can see when there are expansions. For example, if you map several gene trees to the species tree, you will see like a line of several duplications at the same point, right? Like the merging of duplications.